Hello, welcome to Biology Lesson 2.05, the polymers of carbohydrates. You'll notice that we are on a different page. We're on page 6, underneath the line item that says create a Venn diagram for the 4 categories of biomolecules. The space beneath this was supposed to be for that activity, but I realized you're missing a page out of your note packet, so we're going to have to squeeze in some notes other places. So to start off with, we're going to talk about the polymers that we make from the monomers of carbohydrates. And we really only talked about one monomer for carbohydrates. Do you remember what that was? Well, the correct answer was glucose. Hopefully you said glucose. So the monomer that we're working with is glucose. And we're going to talk about 3 polymers. The 3 polymers of glucose, 2 of them are in plants. So we have, let's start off with, and I'll make it green since we're talking about plants. These are carbohydrate, of course. So plants, where do plants get their glucose from? Turns out they get it through photosynthesis. Let's write that down. And they do 2 different things with this glucose. They snap it together 2 different ways. So the first polymer that we're going to talk about, glucose in plants, is starch. And starch are glucose, and I'm going to do a lot of little G's, and I'm going to hook them together here. And then I'm going to connect them with bonds. So starch are these long chains of glucose. Well, my pen will work. Hooked together, and they're in fairly straight lines, not perfectly straight. I'm going to try to autofocus here. I feel like I'm a little out of focus. All right, that's about as good as we're going to get, I guess. But why would we want these in ready, accessible straight lines? Well, starch is how plants store their glucose that they're going to use. So you guys might think, okay, they made their food, they're done. Well, just like us, we're going to learn they have to take this glucose and they have to turn it into ATP. To do that, they need glucose that's going to be pretty accessible. And so we looked in the last video on dehydration synthesis and hydrolysis. If they want to store the glucose, they'll snap those together through dehydration synthesis. And if they need to access it, they'll start cracking these off with hydrolysis, sending them to mitochondria to make ATP. But again, this keeps it pretty simple. And you've heard of starch before. So you heard of starchy foods. You're probably talking about potatoes or rice or grains. Guys, these are all plants. And the reason we call it starchy foods is because the molecule, which is a polymer of glucose, is actually starch. The other polymer that plants have of carbohydrates is called cellulose. And I'm just going to make my little glucose as circles this time, and I'm going to show you how cellulose is different. Let's hook these together. I'm going to put another chain right next to it. I'm going to hook those together. So we have these long lines of starch lying next to each other with cellulose. But then what the plant does is it cross-links. This is called when you make a bond between 2 big molecules. So this is a long molecule and this is a long molecule, but they're hooked together side by side, and we have lots and lots of these, and they're all hooked together. Well, if we needed to crack something off with hydrolysis, would it be easier to do it from here or from here? And the answer is it would be much easier to do it from here. So starch is the usable glucose. to make ATP. But cellulose is tough, it's hard to break, and this is what the plant uses for structure. Might have cut off there a little bit, structure. If you've ever eaten a piece of celery and you took a bite into the celery and you pulled and the strings came off, like there's strings on the back of celery, that's cellulose. And that gives you an idea of how tough it is. If you eat a lot of celery, you know it when you go to the bathroom the next day because you can look in the toilet. It's a little bit gross, but it's true. And you can go, oh, look, it's the celery strings. Well, why does that happen? We can't digest this. We can't break these bonds. It's too tough. When we are at school, we'll usually go outside and play Carbohydrate Red Rover. I'll have everybody stand holding hands, and that represents starch. And I'll have somebody come ripping across the yard, and they'll break through, and they can snap through these bonds no problem. And then we play Carbohydrate Red Rover, where everybody links arms at the elbows. Now, when someone tries to break through, they can't get through. And that's a good analogy for the toughness of cellulose versus starch. Well, that's in plants. Let's talk a little bit about animals. So, in animals, the polymers of carbohydrate, we only have one, and it's called glycogen, Glycogen, glycogen. And your particular stores of glycogen, or your dogs, or your cats, or a frog's, are in the liver. So this is stored in the liver. And it's a little bit like, I'm going to go ahead and write over here. Let's draw the structure. So I'm going to make dots. And each of these dots is going to be a glucose. What happens is, as you eat your spaghetti for supper, I'm going to go to black. We get dehydration synthesis starts to hook together all your little glucose molecules. And then off of here, you might put a branch. And then over here, you might put a branch. We're going to branch out here. I'm going to branch out here. I'm going to go here. Gosh, this is easier to see. Maybe I should write from right to left. So you get all of this branching structure, and each of those little circles is a glucose. Then when you need them, like maybe you're running a cross-country race, your body will do hydrolysis and it will crack off these glucose and start to use those to make ATP. What are we using our glucose for? The same thing. So it's stored in the liver. Glycogen is stored in the liver, and it's used for carbohydrate storage. And we're going to say short term. And our glucose is used for the same thing. Used to make ATP. The interesting thing is each one of these glucose molecules has 6 water molecules around it. So if you're running a cross-country race and you weigh yourself and you weigh 150 pounds before the race, it would not be unusual if after the race you weigh 145. Well, you sweat a lot, so you lost liquid there. But the other thing is you're using these glucose like crazy. You're snapping these guys off, sending them to mitochondria to make ATP, and you're just going nuts. Each one of these glucose has 6 water molecules around it. So not only are you losing the mass of your glycogen, you're also losing the mass of all of that water. So, or if you've ever been on a kind of a crash diet, you might have noticed you lost a lot of weight those first couple of days. That's water weight, and that's because of the way that our body stores glycogen. So, what are our takeaways? We're really only talking about the monomer of glucose, and we're talking about how it's stored in plants and animals. In plants, we have 2 choices. Their readily available glucose to make ATP is stored as starch. For structure, and this is also in plant stems, we have this tough, hard to break cellulose that has these bonds in between. In animals, in the liver, we have glycogen, It's got these big branched molecules, and this is a great example. These are great examples of macromolecules. So when we're talking about macromolecules, we're talking about starch, cellulose, glycogen. We could have uh, tens of thousands of molecules of glucose in here. And this is also for short-term energy storage. Thanks for watching and have a great day.